Hello students, we continue with the topic of photosynthesis today. Now we are familiar with this particular reaction, that is 6CO2 plus 12H2O in the presence of light and chlorophyll gives us C6H12O6 plus 6H2O plus 6O2. Today we know this reaction very well, but then before the 1930s this reaction was not known. Also, it was believed that during photosynthesis, carbon dioxide splits into carbon and oxygen. So, they used to imagine this type of reaction taking place. That is, carbon dioxide would give carbon and oxygen during photosynthesis. Oxygen would get evolved and carbon would then react with water, forming carbohydrates. This was what was the reaction which was imagined to take place at that time. Later on, by the experiments of various scientists, it was proved to be wrong, and today we know what takes place during photosynthesis. We are first going to study Van Niel's experiment today. Now, Van Niel was a Dutch microbiologist. In 1931, he was working with purple and green sulfur bacteria. These bacteria are photosynthetic in nature, and their unique reaction is that they use hydrogen sulfide instead of water for photosynthesis. See, green plants use water for photosynthesis, but these bacteria, they use hydrogen sulfide for photosynthesis. Now, Van Niel observed that when these bacteria are carrying out photosynthesis, they evolve sulfur instead of oxygen. So, over here in the reaction, sulfur is evolved and not oxygen. So, this was the reaction which he observed at that time. The equation becomes 6CO2 plus 12H2S. Remember, hydrogen sulfide is used by green and purple sulfur bacteria. Gives C6H12O6 plus 6H2O plus 12S. Now, because sulfur is evolved and not oxygen, Van Niel further said that in green plants also something similar must be happening. See, he observed that during photosynthesis by green and purple sulfur bacteria, hydrogen sulfide is getting split up. Obviously, hydrogen sulfide is getting split up into hydrogen and sulfur, and the sulfur is what is getting evolved during the process. Sulfur is getting produced during the process. So, Van Niel said that during photosynthesis in green plants, carbon dioxide must not be getting split. Rather, water which the green plants use for photosynthesis must be getting split up into hydrogen and oxygen, and that oxygen must be getting evolved. So, he was the first scientist to state that water is used in place of hydrogen sulfide and oxygen is evolved in place of sulfur during photosynthesis by green plants. That means carbon dioxide is not split during photosynthesis. Now, Rubin, further another researcher named as Rubin, he proved this reaction by his experiment. Now, Rubin in 1941 observed photosynthesis in green algae, that is chlorella. He used water which was labeled with heavy oxygen, that is the water contained O18, the isotope of oxygen, that is O18. Remember what are isotopes? Yes, isotopes are atoms having the same atomic number but different atomic weights. For example, the normal isotope of oxygen is O16. The heavy isotope of oxygen is O18. It is oxygen only because the atomic number is same, but atomic mass varies. Atomic weight varies over here. So, Rubin used water labeled with heavy oxygen. He used water which contained O18. So, H2O18. He observed that during photosynthesis by chlorella, which used this particular water, the oxygen which was evolved was O18. Now, this proved that oxygen which is evolved during photosynthesis comes from water and not from carbon dioxide. The oxygen does not come from carbon dioxide, rather it comes from water. Thus, this reaction we get for photosynthesis. Rubens reaction we get this way, Rubens equation, 6CO2 plus 12H2O 18 gives us carbohydrates, that is glucose, plus water plus oxygen, which is heavy. And this is how it was proved that oxygen comes from water and not from carbon dioxide. Now we come to a classic experiment performed by Robert Hill, which conclusively proved that in photosynthesis water is split up into hydrogen and oxygen. Now this is known as Hill's experiment. Hill took spinach leaves and isolated chloroplasts from the spinach leaves. Remember, chloroplasts are the organelles in which photosynthesis takes place. Now you have to remember certain facts about this. Since chloroplasts were taken out from the spinach leaves and put into a test tube, taken in a test tube, that means, and suspended in water actually, that means what? There was no carbon dioxide in the experiment. Carbon dioxide was not supplied to the chloroplast. Why? Because chloroplasts get carbon dioxide through the stomata of the leaves. Now these chloroplasts have been taken out from the leaves and therefore there is no carbon dioxide in the experiment. Now he also added a synthetic electron acceptor, that is ferricyanide. So ferricyanide is a compound which acts as an electron acceptor in the experiment. Now remember, anything which accepts electrons, an electron acceptor is a compound which can get reduced. Okay, one definition of reduction is accepting of electrons. Also accepting hydrogen, the same thing, accepting electrons is called as reduction. So ferricyanide was the electron acceptor in the experiment. He took this apparatus and exposed it to light. After some time, he found that there were bubbles of oxygen which were evolved. Now from where did the oxygen come? He started thinking, from where did the oxygen come? Because there was no carbon dioxide in the experiment. And therefore carbon could not have been split into carbon dioxide could not split into carbon and oxygen. That means there was only one conclusion, that the oxygen must have come from water. The leaves, the chloroplasts were suspended in water. So the oxygen must have come from water. That means water molecules split up into hydrogen and oxygen. Now how do we know that hydrogen was there? How do we know that water molecules split into hydrogen and oxygen? We know this because at the end of the experiment, not only did he find oxygen bubbles getting evolved, he also saw that ferricyanide was reduced to ferrocyanide. Reduced form of ferricyanide is ferrocyanide. So electrons were accepted by ferricyanide and thus at the end of the experiment ferrocyanide was formed. So this experiment could take place only in the presence of light. If we kept this apparatus in the dark, there was no evolution of oxygen and no reduction either. That means in the presence of light only, water can split up into hydrogen and oxygen. And thus this was known as photolysis of water. Lysis means breakdown, photons light. So breakdown of water in the presence of light is known as photolysis of water. So Hill's experiment proved that during photosynthesis water gets photolyzed and thus oxygen is evolved. So the classic Hill reaction is what I have written over here. It is 2H2O plus 2A. 2A is the unknown electron acceptor. Is the compound which will get reduced during the reaction. At that time, Hill did not know which is the compound in plants which accepts electrons. And therefore, it was just written as 2A, an unknown compound. In the presence of light and chlorophyll gives 2AH2. As you can see, the unknown electron acceptor has become reduced in this case. This compound has become reduced and plus oxygen is evolved during the process. So this reaction is known as the Hill reaction. So from the Hill reaction or from Hill's experiment, we see that in photosynthesis, oxygen is released from water and electrons for the reduction of carbon dioxide are obtained from water. So these electrons which are evolved, they first go to the unknown electron acceptor, and this unknown electron acceptor in turn passes them on to carbon dioxide, which gets reduced. I hope you are understanding the concept over here. Now, what was the unknown electron acceptor? Further, Hill could not find the unknown electron acceptor. It was Arnon, another researcher, who found that the unknown hydrogen acceptor or electron acceptor in plants is the compound NADP. NADP stands for nicotinamide adenine dinucleotide phosphate. And this compound during photosynthesis gets converted into NADPH by accepting hydrogen. And this hydrogen is then passed on to carbon dioxide. And thus carbon dioxide gets reduced to form carbohydrates. Thus we get sugar getting formed or glucose getting formed, which is a carbohydrate during photosynthesis. And at the end of the reaction, we get back NADP, which will again accept hydrogen, and the reaction will go on. So that is how the entire reaction regarding photosynthesis was found out. These were the earliest experiments which were performed. Now, thus we come to the modern concept of photosynthesis. So today we know that photosynthesis is a redox reaction. Now, what is the meaning of a redox reaction? A redox reaction is a reaction in which reduction and oxidation as well as oxidation takes place at the same time. It involves oxidation as well as reduction. Now, during photosynthesis, what gets oxidized and what gets reduced? As you can see, water gets oxidized. See, oxidation, one meaning of oxidation is removal of hydrogen. So water gets oxidized, hydrogen is removed from water, and thus we get oxygen formed. On the other hand, whatever hydrogen is removed from water ultimately goes to carbon dioxide, which gets reduced, and thus we get glucose getting formed, carbohydrates getting formed. So in this way, photosynthesis becomes a redox reaction. Now remember that this reaction, the oxidation of water into oxygen, requires light, and therefore it is known as the light reaction. So this reaction is light dependent reaction. It requires light. So it is called light dependent. And this reaction is called light reaction. On the other hand, reduction of carbon dioxide does not require light. It is a light independent reaction. Light independent reaction. Now the light dependent reaction takes place in the thylakoid of the chloroplast, on the thylakoid membrane. Yesterday in the previous lecture we learnt about thylakoids. So this takes place in the thylakoid membrane, on the thylakoid membranes, that is there. Whereas the light independent reaction takes place in the stroma of the chloroplast. So that is how we learn quite a bit about photosynthesis because of the experiments which were performed by earlier scientists.